Welcome back to Notre Dame Day. I'm Pete Byrne, and with us now is Jeff Schorey, a professor in the College of Science, who is the co-author of a study that made a landmark discovery in the treatment of tuberculosis. He's here now to tell us all about it. Jeff, welcome to our program here. Tell me about your study and what it is that you discovered.
Well, first, thanks for inviting me.
Pleasure.
So I kind of do a little bit of background. So TB is not something that a lot of us think about here in the United States, but it's a major problem worldwide. It's actually the #1 cause of death by an infectious organism. About 1.6 million people die of TB a year. So that's obviously a large number. So the kind of question is, why does it kill so many people? And there's a few sort of reasons for that. One, we don't have a good vaccine. Second, our diagnostic tools are not the best. And third, the drug treatment itself is long and takes a lot of antibiotics. It's 6 to 9 months and 2 to 4 antibiotics. So you might imagine the compliance is pretty poor.
Right.
And if you don't take your antibiotics, it leads to an increased chance of drug resistance. And that is something that obviously is a problem globally. As far as my lab and sort of the study, we're interested in understanding pathogenesis. What makes this bug so successful? And we've been specifically looking at what we call these little membrane vesicles. They're like bubbles. But they're really, really small. They're about the size of a virus. And it turns out that cells release these vesicles as a form of communication. So one cell would release the membrane vesicles, and it will be taken up by another cell, and the cell would respond to that. And they might, for example, undergo cell proliferation. So kind of had the question of why does this have anything to do with TB? Well, it turns out that Mycobacterium tuberculosis is not only infects you as a person, but it also gets inside your cells. It's an intracellular pathogen. So we're interested in how it gets into the cells and what's the response. And it turns out that some of the mycobacteria components that the mycobacteria have are released, and they get into these membrane vesicles, and these membrane vesicles are released from the infected cells, and then they act on other cells of the immune system. So it's a way for the immune system to be alerted to the infection, to the bacterial infection. And so we've published a number of studies sort of looking at how they form these membrane vesicles, what these membrane vesicles have as far as mycobacterial components, also sort of what it does to the host immune response. This paper specifically, the one that we published recently, which was in EMBO Reports, looked at what these vesicles do in the context of RNA, the nucleic acids that's present in them, so what they sort of do there.
So for those of us who aren't chemists or biologists, how would you in layman's terms describe why this discovery is significant?
So, so these membrane vesicles, what we found is, and I don't know, I may be showing my age here, but Popeye. You ever know Popeye?
Yeah, I do. Maybe showing my age, but
yeah,
yes.
Yeah, Popeye. So Popeye, he's pretty wimpy, right, when he doesn't have his spinach. But if he takes his spinach, you know, he's tough. He's tough. So sort of the analogy works here, where there is a host cell that takes up the mycobacteria, and that mycobacteria it wins. It kills that cell, multiplies, and moves on. Turns out that these vesicles, if you treat the host cell with the vesicles prior to the infection, those host cells are now able to control it, like the spinach for Popeye. They're better able to control the infection. And so that's something that we think has a distinct advantage to the host response.
I know you guys have taken this to trial. It's just tested positively on mice. Is there a human trial possibly in the future?
Well, so what we've done is, so the idea is how can you perhaps use this in humans, right? And it turns out that there's a type of approach that's used a lot in cancer called immunotherapy. Probably you've heard about. Millions of dollars is spent in this area. And the idea is that you would treat with chemotactic drugs as well as these sort of immune stimulatory drugs. And the combination of the two of them actually would promote the removal of the tumor. So the idea is, can we do the same thing with infectious diseases? That is, take an antibiotic and take one of these immune therapeutic drugs, like these vesicles, combine them together and do something better than what you see with the antibiotics alone. And so what we did in this study is we sort of did a mouse study, as you mentioned, where we combined these vesicles with an antibiotic, and we found that those combination was much better than either of them alone.
Okay.
So we think that the combination will do well.
Well, Jeff, it's fascinating stuff. We're out of time, but I want to thank you for coming on Notre Dame. Thank you.
Sure.
Thank you for the good work you're doing here at Notre Dame and also in the world, and we look forward to seeing how this process continues. So thank you.
Thank you.